Welcome back, everybody. We've got 3 speakers now between now and lunch. So first speaker is Duncan Barlow. He's going to come up now, and I'll get him to introduce himself and do the first speak.
Morning. My name is Duncan. As I was introduced earlier, my LinkedIn profile says I'm a microbial identification enthusiast. My real job title is I'm a technology and market development manager, so I do have a real job. Okay. I work for Charles River Microbial Solutions. Charles River supply QC microbial solutions to the pharmaceutical industry. So that covers endotoxin testing, rapid microbial detection, and identification services. And I work specifically in the division that provides microbial identification services. So in this talk, I'm actually going to describe a case study that was shared with us by one of our customers. I'm going to have a look at their investigation they had related to an out-of-spec that they found. I'll then go into looking at different microbial identification methods and their impacts and costs to companies. And we'll finish up with a quick summary, and then if anyone's got any questions about microbial identification, I'll be more than happy to take those. So in the case study, it actually started with an endotoxin out of specification. It was an out of specification event in a semi-finished product, which was packaged in a plastic 500 ml ampule. Now, as I'm sure you're all aware, endotoxins are pyrogens. Endotoxins are made of lipopolysaccharides, and lipopolysaccharides come from the cell walls of gram-negative bacteria. And of course, if it remains in the product and ends up in the patient, it can cause serious harm to the patient. So when an event such as this occurs, obviously it has to be investigated. And when investigating an out-of-spec for endotoxin, typically the culprit you're looking for is a gram-negative bacteria, because that's where endotoxin comes from. So just to go through the initial response to the out-of-spec, an investigation was opened for the deviation. There was the routine bioburden testing revealed there was growth below specification, but it was noted that the growth was not common, it was out of trend. So to help understand what was happening, they decided to identify the bioburden. Now, this company had an in-house ID system, which is an automatic phenotypic ID system, which is quite a common ID system for people to have in-house. And the way that system works is you have to carry out a gram stain before you put the sample onto the system, and the gram stain tells you which type of reagents you use for the next steps. Okay? They identified their bioburden as a bacteria called Cocuria cristinae. Okay? Now, Cocuria cristinae is a gram-positive coccus, so it's a gram-positive, so it's not a gram-negative. It's typically associated with humans. We've heard a couple of speakers this morning talk about the sorts of organisms you find in clean rooms. Most of them come from humans, so it's not unusual to find a gram-positive. But the out-of-specification was for endotoxin, which was a gram-negative, so there was a little bit of a disconnect there. So they should have had some concerns at this point, that there was a little disconnect. They had what they suspected was gram-negative, they identified it as a gram-positive. I'd just like to point out, the identified organism is Cocuria cristinae, but bacterial taxonomists like to make things difficult for people, so that species was actually reclassified to a completely different genus back in 2018. So you have to be aware that things may change their name as well when you look at names. So just going through the investigation, there was a lab investigation, as you'd expect. And during that investigation, they determined there was no contamination by the analyst and no lab error was detected at all. They also carried out a comprehensive environmental monitoring review, and they found nothing of significance that could help with their current investigation. They did find some occasional problems which are more than 10 years previous, but they were deemed to be unrelated because they were so long ago. The batch that was associated with out-of-spec was quarantined, pending the results of the investigation. So a critical CAPA, corrective and preventive actions, was opened. It involved their head office because this was a batch that was on hold, so it was a serious case for them. They held meetings every 2 weeks to provide updates on the investigation and the actions taken for the possible causes of the out-of-spec. The production line was stopped, the machine and the heat exchanger were dismantled, maintenance was carried out, and they did chemical and heat sanitization on those bits of equipment. The production line was restarted with extensive sampling at the most critical sample points, which were identified by the production team. The progress of the CAPA was slow because they were working around production scheduling and they were trying to manage the associated costs with having a line not working and all the team and people that were involved in it. And they estimated that the CAPA was going to be open until the end of the following year. So several months this was going to be open whilst they worked on determining the root cause and what the most appropriate CAPA was. What was the root cause determination? Well, unfortunately, they couldn't determine a root cause. Okay? They were suspicious of the heat exchanger and the cold water that surrounds it, but they changed the heat exchanger, as I said, and after evaluation of the unit, they found nothing to indicate that it was the cause of their issues. So they actually implemented some corrective actions, as we saw on the previous slide, and it can be a good idea to do something when you've got a batch on hold and a production line stopped. But when you don't actually know what the cause of the problem is, sometimes these actions can be, they can make no sense. They're not going to fix the problem you have. And it can be even more problematic if you implement that CAPA into SOPs and there's ultimately no benefit to you with respect to addressing the problem that you've had. Same customer, fast forward 2 months, guess what happened? Another semi-finished product presented with an out-of-trend microbial count. Again, they used their in-house phenotypic ID system, and again, they found Cocuria cristinae. It was found on the same filling line and the same product as the previous out of specification. So the investigations were linked as it was from the same product, same filling line, and the same identification. So they recognized that their initial problem had never been fixed. This time, though, there was no endotoxin out of specification. It was a bioburden out of trend again. So something's going on here. They're not getting to the bottom of what's going on. So what did they do next? Well, they reached out to us at Charles River for help. They were having conversations with us about endotoxin testing, because we supply endotoxin testing products, and they mentioned this investigation and what they were trying to achieve. So when they spoke to our technical specialists, they shared the gram stain images that they'd generated in-house. They shared that with our Acugenics ID specialists. And they told us what ID they'd got from their in-house system. So they shared that information with us. Now, we suggested that they send in their sample, their isolate, for DNA sequencing using a 16S validated sequencing service to help shed light. Sequencing can be the best way to get your most accurate result. Unfortunately, they only had the isolate from the newest out of trend because the original isolate had been discarded, so we only had the second one. So we ran it through our 16S ID service. And with our sequencing service and our validated database that we use, we identified the organism as Acinetobacter nosocomialis. Okay? Acinetobacter is a gram-negative coccobacillus, so more of an oval shape than either a circle or a rod. You can see here this is the phylogenetic tree identifying the isolate. So when we do DNA sequencing for our customers, we take the sequence and we can visualize evolution looking at the relatedness of the isolate to the other entries in our library. The horizontal dimension indicates the relatedness, and you can see there's no difference between these two. This is the sample number, and we've redacted the sample number because it's unique to this customer. And you can see that it exactly matches with Acinetobacter nosocomialis with a 0% difference to the library entry. So we are very confident that we have ID'd this correctly. Now, Acinetobacter is an environmental organism. It's an opportunistic pathogen. It has caused hospital-acquired infections in humans. But as I said, it is not gram-positive. So remember, when they sent us the isolate, they thought they had a gram-positive Cocuria species. So their original ID appeared to be incorrect. And we'll take a look at why that was in a few slides. And the importance of the ID, especially during an investigation, can't be stressed enough. An investigation for a gram-positive human-associated organism will look very different to an investigation looking for a gram-negative environmental isolate. So with this new information, the team refocused their investigation on the heat exchanger again, since Acinetobacter and gram-negative rods in general are associated with sources of water. So they suspected there was something going on with their heat exchanger, but they couldn't get to the bottom of that problem. But they went back and revisited it. They performed additional environmental monitoring, and they found another isolate, which they identified as Cocuria varians, which is also a gram-positive. It's the same genus, or it was the same genus as the original sample. But we actually found that that ID was wrong as well. It was actually a Corynebacterium species, which is a gram-positive rod using our DNA sequencing service. So again, their in-house ID system had given the wrong name. The first time it gave Cocuria cristinae, this time it gave Cocuria varians. It was two different organisms. The first one was actually an Acinetobacter, the second one was a Corynebacterium. And if you think about that in terms of just day-to-day monitoring, you're trending. If you've got an ID system that gives inconsistent results or incorrect results, how can you trend accurately if your ID results are wrong? They also found an organism called Stenotrophomonas multifila, which is also a gram-negative, so again, potential for the source of the endotoxin. We identified that for them using DNA sequencing, but they never found the Acinetobacter in this second out of trend that they had. Unfortunately, they had no further out of trends or out of spec events, and they decided to destroy all the batches that were associated with these out of trends and out of specs. So, quite extreme thing. Batches can cost an awful lot of money. Could it have been a better investigation if their IDs had been right the first time? Potentially. So as I said, the batches were discarded. So what actually went wrong? We saw their ID system gave incorrect results, at least on 2 occasions that we're aware of. Why is that? Well, as I said, the phenotypic method they used requires a gram stain prior to analysis of the sample on the system. I don't know if you're familiar with the gram stain. The gram stain is a manual procedure. It requires human interpretation. It can be error-prone and subjective. It needs very strict timings, and I think there's about 14 steps in that assay. So you can introduce timing errors, for example, at any point in any one of those 14 steps. But it's not just as simple as having better trained analysts. This is the image on the left is the gram stain that customer sent us. Anyone familiar with gram stains? Anyone want to hazard a guess at what that looks like? It looks like a gram-positive cocci, which is what their ID system told them it was. So, looked good. That looks like a gram-positive cocci to me. Okay? But when you zoom into the image, this is actually not a zoom in of this image. This is taken from an article just to stress the point, because the picture quality wasn't good enough to zoom in, unfortunately, to show you what was happening. This is a gram stain of an Acinetobacter. Okay? Now, you can see the one with the arrow next to it. It's a little bit more of an oval than a nice circle. Okay? And that indicates how difficult it can actually be when you're looking down a microscope to say, is that a circle or a rod? Some rods are nice and long and really obvious. Sometimes they're really short, it can look just like a cocci. But what's even more striking is the color. The gram reaction is the color. This looks like a gram-positive reaction to me. It should be pink if it's gram-negative, but this is a gram-negative bacteria. Okay? Now, the reason for that in this case is Acinetobacter are actually notoriously difficult to destain, meaning they retain the stain that should stay in the gram-positive, and they don't turn pink with all the subsequent steps. So they look like gram-positives. So there's only so much training you can give an analyst when they're doing this assay. Microorganisms don't always play ball. They don't like to conform to our little rules. Okay? This genus is notoriously difficult to destain. Okay? So when you're doing a manual subjective technique such as this, how can you be sure you've always got the right result? Yes, you can. Some things are really obvious when you gram stain them. Really obvious what they are. But there's a lot of organisms that aren't. Okay, so if anyone's interested in reading more about it, the link to the article which discusses the issues with gram staining them is there. I believe everyone gets the slides after the conference. So the gram stain is the first step in the process flow for that phenotypic method that customer used. So if they get the gram stain wrong at the start, they're always going to get the end result wrong at the end. Okay? There's no way to correct that. It's always going to give you a gram-positive result at the end. But it's not just the gram stain that's the issue here. We saw in the case study there was 2 wrong isolates. One we identified as a gram-negative, which their system got wrong. The other one we identified it was a gram-positive. Their system still got it wrong, but it was a gram-positive, which means they did the gram stain correctly, but it still gave the wrong result. So we can't just say, oh, this is an issue with the gram stain. This is a diagram of sort of accuracy and reproducibility of microbial identification methods. So broadly, you have genotypic methods, proteotypic, MALDI-TOF, and we've heard it mentioned this morning, I think in the first talk, and the phenotypic methods. Okay? Genotypic methods are regarded as being the most accurate ID method. And that's because you're comparing DNA sequences from your unknown to a database of known sequences. So it has the potential to be the most accurate, and I'll come on to why I say potential and not just say it is the most accurate. There is another sort of side topic here. When you're identifying bacteria, the gene target you use is straightforward. You use the 16S part of the gene. That's widely accepted. It's what everyone uses for microbial identification. It's not a problem. But for fungi, there's actually different gene targets you can look at. You can look at the ITS2 region or the D2 region. Okay? So depending on which gene region you choose for your fungi, can affect the accuracy of your results. Proteotypic, the MALDI-TOF, whilst potentially not as accurate as genotypic tests, they can still be highly accurate and reproducible. And that's because they're analyzing highly abundant proteins that are consistently expressed in the microorganisms. So, for example, ribosomal proteins. One of the speakers earlier mentioned about how easy it was to run a MALDI-TOF system. It's fast. But unfortunately, MALDI-TOFs are expensive. It requires significant capital expense to invest in a MALDI system. But they are nice, accurate methods with some caveats. Then we come to the phenotypic ones. These are still quite commonly used. I would say a lot of people are starting to move away from them towards more the MALDI technology. And phenotypic methods encompass things such as the gram stains that we've discussed, and biochemical analyses such as oxidase tests, catalase tests, and so on. Now, as I said, the gram stain is very often the very first step in a phenotypic analysis, and the result of that test tells you what to do next. Get it wrong at the start, your result's always going to be wrong. We saw from the case study, the first incorrect ID was down to a really difficult gram stain to interpret. It was just a tricky organism to do a gram stain correctly. The second one wasn't. The second one, they got the gram stain right, but the ID system gave them the wrong name. Okay? And the reason for that is, for a phenotypic method to give an accurate result, it needs to be in a happy, healthy state. It's about expression of its genes. Now, when you isolate things from a clean room, clean rooms are designed to stop bugs from being there. They're low nutrient environments. So bugs that have been isolated are often stressed. They might have been exposed to cleaning and disinfection products. They might even have been exposed to pharmaceutical products. So they very often don't react as you would expect them to. So phenotypic is regarded as being not the most accurate way of generating an ID. And I think even in the pharmacopoeias, the US pharmacopoeia, it mentions about how genotypic methods are more accurate and precise. Might be the FDA guidance, actually, but it's recognized that genotypic methods are the most accurate way of doing it. One thing I would just like to add as a sort of side note to this, these technologies are used commonly for routine ID. But there are some groups of organisms that are so closely related, that they are different species, that they can't be distinguished with MALDI or even sequencing. Not the routine sequencing that I've mentioned, 16S and ITS2. Okay? And I'll come on to that. So an accurate ID, where does it come from? Well, as I've discussed, the technology plays a really important role. You have to select the appropriate technology for what you're trying to achieve and the criticality of your sample. But there's a second part to it. Your ID system is analyzing data. It's making a judgment call. So, for example, I've listed here the match factor. In MALDI, the software does a calculation of how closely your unknown protein spectra matches the protein spectra in the library, and it does a calculation and says how closely that matches and sets a cutoff value. Now, you need to be aware of what that cutoff value is. What confidence is it giving you in your result? Is it a confident species ID? Is it a low confidence species ID? What does low confidence species ID even mean? Can you use that? I don't know. And there's also, when you look at the match factor, there's a trade-off between getting high rates of ID, so IDs for every sample you put on it, versus the accuracy. If you increase the accuracy, you get more and more no IDs. So you have to have that balance of the analysis to generate the match factor that has to be correct. For sequencing, I showed you the report we generated, phylogenetic tree. It's important to be aware of this because you can buy automated sequencers that will give you a comparison to the unknown. It tells you how closely related your unknown is to the library entry, and it will say it's 0% difference, for example, or 0.1% difference, or 1.2% difference. Now, there's no universal cutoff for species call in sequencing. Okay? It can vary by genus, it can vary by species. Okay? So it's really important to look at that phylogenetic tree because it takes into account the relatedness and the evolution, so you can actually see visually where your unknown is compared to the library entry. Okay? I can't stress this enough. This is probably the biggest thing. The technology you select plays an important role. The analysis is important. The library that comes with your system is critically important. A library is affected by whether or not the organism you're trying to identify is present in that library. Okay? You cannot ID something if it is not in the library. Okay? The library is affected by the diversity of the organisms, and that's especially important in MALDI. Okay? With MALDI, the way it works is because you're looking at protein spectra from your isolate, within species, different strains have slightly different protein spectra. And just because your MALDI library has, say, Micrococcus luteus in it, it's not a guarantee it will always identify Micrococcus luteus because there are different strains, you get strain variability. So the library needs to have good strain representation. When you actually look at the MALDI libraries, you'll see they talk about like main spectral patterns. There might be 10 to represent a species. There might be 40 to represent a species. And you need to have that diversity for MALDI, and the data that makes up the library needs to be accurate. And I mentioned things like name changes, the taxonomy needs to be correct. And all of that combines to give you the most accurate result possible. Just quickly, I mentioned very closely related species. There's a number of groups of species that are indistinguishable by MALDI or sequencing. So this is an example of the Burkholderia cepacia complex, but it's true of other groups, such as the Bacillus cereus group. And this is a phylogenetic tree, and you can see there's very little difference, horizontal difference between all of these entries in that group. And this is looking at 16S. So you can't say which species that is looking at that tree. The best you can say is it's Burkholderia cepacia complex. But If you need to know which species, there are alternative gene targets. So if you look at the REC-A gene, you can see this much more defined horizontal difference between all the entries. Okay? So just a side note to be aware, when you're looking at a critical isolate, if it's something that's closely related, you may need to look at advanced techniques to actually be able to identify that correctly. And we saw the case study. They were having an ID in an investigation, and they're looking to find the root cause. Where did that contamination come from? How did it get there? But what if your isolate is, if your contamination is caused by a very common organism? So something like Micrococcus luteus. Typically, everyone sees that in their top 10 most frequently occurring organisms, probably position 1 or 2. So if you recover a critical isolate, which is something like Micrococcus luteus, and you're trying to track the source to prevent that issue from happening again, you're going to see that organism in a lot of your samples. It's so common. So as an example, if you have this critical isolate and you do more environmental monitoring and you find that same organism in 32 different places, how do you know which one's the likely source of the contaminant? You can't. You can, actually. You can use strain typing. Strain typing looks at different gene targets within the genome of the isolate you have and can actually compare them. So this is all genetic sequencing of Micrococcus luteus, different samples. And you can see here, these two match exactly. So the critical isolate is the one ending 3,9, and another example of Micrococcus luteus, this one here matches it exactly. So it's likely to be the source of their contamination. So it's an advanced technique that can help in investigation cases like we saw at the start. So there's different techniques available. Some are more accurate than others, but what are the impacts and costs of inaccurate ID? I'm sure you hope to get away with someone that wasn't going to mention Annex 1, so I apologize. But I'll just quickly go through this. Jim mentioned earlier contamination control strategy. He spoke about the foundations, one of the foundations being scientific knowledge. Okay? ID systems give you scientific knowledge. But there's also other elements that are listed in here, cleaning and disinfection. It talks about using scientifically sound methods to detect contamination. It talks about prevention mechanisms such as trending, investigations, root cause determination in CAPA, which we saw from the case study. Looking at what it says about disinfection, it says monitoring should be undertaken to assess the effectiveness of the disinfection program and to detect changes in types of microbial flora. For example, organisms resistant to the disinfection regime currently in use. Jim showed some really nice examples of how different species of fungi can have different resistance to different organisms. So it's really important to know what you have present. It's a requirement to know what you have present to ensure that your cleaning and disinfection remains effective. Very clearly, in section 931, anything detected in grades A or B area should be identified to species level. So that's really straightforward. Okay? If you've got an ID system that gives genus-only results or low-confidence results, how do you get to species level? But it actually says the potential impact of such microorganisms on product quality and overall state of control should be evaluated. So your ID needs to be accurate. And it also says consideration should be given to identification of organisms detected in C and D areas. And again, it talks about things like organisms that are difficult to control, such as spore formers, and you need to understand the typical flora of these areas. So when you're looking for that understanding, you need good scientific data to provide that knowledge. So your ID system needs to be able to provide accurate IDs. Trending's mentioned, changes in microbial flora type, predominance of specific organisms, spore formers and molds again. Okay, so again, you need accurate ID to be able to do trending accurately. And just as an example, this is a graph. So at Accugenix, we identify hundreds of thousands of isolates for our customers each year globally. And this is the top 5 fungal species our customers send us for identification. Fungi can be problematic. As mentioned earlier, they're spore formers that can move around in the air. But fungi are also metabolically diverse. They can affect the therapeutic properties of a product, a non-sterile, for example, if they're in there. And obviously, spore formers, if they get exposed to moisture, they'll proliferate. You can see on the graph there's a very clear trend into the autumn or fall months. And that's because there's a lot more of them in the natural environment. There's leaf litter, there's harvesting of crops going on, which throws up spores into the air. So the clean rooms will be challenged more at that time of the year. And being aware of these trends allows you to be proactive, knowing that this is going to happen. So ID plays an important role in the contamination control strategy, and you need an accurate ID for that to make sure your assessments are correct. USP 1113, the Microbial Characterization, Identification and Strain Typing chapter, talks about the impacts of IDs. No ID has the lowest impact. You don't know. You have the option to do something else. Use a different ID system. Outsource it. Wrong button again, sorry. Misidentification to species. So it gets the genus name right, but the species wrong. And the biggest impact is completely the wrong name. It gets the genus name wrong. And that leads to things like flawed investigations, looking for the wrong organism in the wrong place. Trending may not be effective. You may have a non-sterile product that you think has no objectionables in it. If your ID system gives you the wrong name, you may release a product that has an objectionable organism in it. And when we think about it in terms of costs, the cost of an ID is fairly straightforward. Most people know what their third-party lab or their in-house system costs them to run. People may even gone to the point of taking into account the labour costs of running an ID system, the equipment costs, maintenance, and so on. But what about the costs associated with misidentifications? If you have to retest a sample because that's either no ID or you suspect the ID is wrong, there's additional costs for the preliminary tests, for labour, the material, and the reagents. But also there's time wasted. When you're in critical situations, you don't want to be wasting time rerunning samples. Misidentifications can also lead to investigation delays. You're looking for the wrong organism in the wrong place, or you've got a no ID, you have to send it to an outsourced partner who takes 10 days to give you a result back. Time is money, especially when you're having investigations, production lines are stopped, so on. You may end up developing an inappropriate kappa. You might not get to root cause, or you might develop a kappa you think will address the issue, and it's the wrong organism you're trying to address. And you may have product disposition errors. Potentially, you could release a product that was contaminated, as I said, like a non-sterile with something in it that shouldn't be there. Or you might destroy a product that actually was fine. You know, most batches of pharmaceutical products cost a lot of money. So these are all hidden costs of a misidentification. So quickly, just to summarize before I'll finish up, and if anyone's got any questions, I'll be happy to take those. If you're going to do an ID in-house and you're spending money on it, you want to be sure the results are accurate and consistent. Or if you outsource it, you want to be sure your outsourced partner is using the appropriate technology and ensuring it can give accurate and consistent IDs. That leads to better knowledge. Okay? Knowledge of your typical microbiological flora. Annex 1 talks about scientific knowledge. It talks about knowledge of typical flora. You get that from your ID program. When you have good knowledge, it leads to better efficiencies. You have more streamlined investigations when something does go wrong. You're more likely to get to root cause and develop the right kappa. Your trending's more accurate. If you know exactly what's present and how it changes throughout the year, it can inform your cleaning and disinfection program more accurately. Ultimately, when you have improved efficiencies, less investigations, more streamlined investigations, when you have kappas that work, it leads to cost avoidance. Investment in a good ID program can help save money in the long run by avoiding these hidden costs that you can get from a wrong ID. It seems like such a trivial thing that ID is wrong, but it can lead to all sorts of problems further down the line. So with that, thank you very much for your attention. If anyone has any questions, I'll be happy to take those.
Thank you, Duncan. Thank you, Duncan, for that. Has anybody got any questions for Duncan? Explained very well. It was very, very interesting for me, very interesting.
If you do, I'm on the booth.
I missed a 5-minute warning because I was listening so hard.
That's all right.
Okay, thank you. Thank you.